I'm Professor William Young of Department of Obstetrics and Gynecology. My research interest is on understanding the molecular mechanism of fertilizations and its relationship with infertility. About 15% of the couples at reproductive age are infertile. And among them, if they undergo assisted reproduction treatments, about 6% of them do have a fertilization problem in assisted reproductions. Our research is on infertilities. Specifically is on the molecules that affects the binding of the sperm to the outer coat of the eggs. And recently, we find that one sequence, sugar sequence on the outer coat of the eggs, and this sequence is responsible for 70% of the binding of the sperm to the eggs. If we block these molecules, 70% of the sperm will not be able to bind to the eggs. And that means this is in fact one of the causes of infertilities. Sperm-egg binding has not been known for many years, although people try to look at it. So far, nobody has been able to identify a sort of sequence that can contribute so much to the binding of the sperm to the eggs. This is the egg, and this is the outer coat. We call it the zona pellucida. And our molecules responsible for the binding is on this outer surface. The molecules will allow the sperm to bind to the zona pellucida, this outer coat of the eggs. In the normal IVF programs, we put the eggs together with the sperm in a culture dish and then allow them to fertilize. For couples, when their fertilization using the standard IVF fertilization technique fail, we need to use a needle to inject the sperm into the oocytes. The procedure is called intracytoplasmic sperm injections, and usually we call it hku. Now we know the molecules responsible for fertilizations, and by identifying couples having defects in the molecules, we can put them into more aggressive treatment like hku here, and therefore increase the efficiency of the whole treatment. Human eggs is very difficult to obtain. We have been able to do this because we have a good IVF programs and we have collaborations from our clinicians that help us to get consent from our patients to get unutilized eggs. Hong Kong U has a very strong scientific background and track records on research in reproductive medicines, including fertilizations, including embryo developments, particularly on the interactions of the female reproductive tract with the sperm as well. We started our collaborations many years ago with Imperial College in United Kingdom. In these collaborations, the Imperial groups helps to look into the structures of the molecules. We use our established sperm function test to look into the functions of the specific molecules.
In this project, we need to collaborate with some scientists in Imperial College, which are experts in mass spectrometry. Mass spectrometry is an ultra-sensitive method which can help us to identify the component of the zona pellucida.
And these proteins, the Hong Kong U team is now looking into the molecules in the sperm that bind to this egg coat. Now with our identified molecule, this can use as a bait to fish out the responsible molecules. And by doing that, we will be able to identify the group of men with potential fertilization problems. We can then know the fertilization better. At the moment, we are in the forefront of identifying the molecule responsible for fertilizations in humans.